Hi, I'm Simin Meydani, Director of Nutrition Center on Aging at Tufts University, and I'm here with Dr. Steindler, the Director of Neuroscience and Aging Laboratory, on a rainy afternoon in Boston, talking about a new paper that was published in Science by Dr. Baruch and colleagues about the role of interferon type 1 cytokines, which includes interferon alpha and interferon beta, and brain function. So Dennis, thank you so much for spending the time to do this. And what I wanted to know is that, what do you think of this work? It seems really very interesting and quite novel. So what is your opinion, and what do you think it does in terms of the whole research area in neurocognition and aging?
Yeah, so this is a game-changing paper, Simin. So till this paper, most of us have been taught for our whole lives of studying neuroscience that these plexi that sit inside ventricular spaces in the brain, called the choroid plexus, is involved in generating cerebral spinal fluid and being an interface between the blood and the brain, where CSF is produced and circulates throughout the ventricle system of the brain. And this flow of cerebral spinal fluid is important for removing toxic substances, for introducing things into the brain, taking brain messages out, and so on. Well, until this paper, there's only recently been some mentioning that the flow of cerebral spinal fluid is actually quite important for cell genesis during adult aging in the brain and just standard operating procedures in the brain. So a sort of new role for these ciliated epithelial cells that sit in the choroid plexi of the brain. But now with the Baruch et al. Paper, what we're being told is that in aged mice, in mice that are, of course, undergoing some sort of viral or other type of infection, the choroid plexus actually becomes a site of inflammation. And along with now other information from Dr. Schwartz's group and from her former colleague, Dr. Kipnis in Virginia, we are now knowing that the T cells that enter the brain in the choroid plexus, as well as in the meningeal spaces of the brain, along with other immune system related cells, including dendritic cells and B cells, that we now have immune system cells involved in inflammation that actually can impair cognition. So I think what we're going to begin to see from this paper now is people beginning to think of immunology in the brain as not just the absence of responses, but the role of responses like this.
No, I mean, I totally agree with you. I mean, when I read this paper, I thought, wow, that's really very interesting, because it kind of really, I mean, we knew of the connection between the immune cells and the brain and all of that, but this really brings it to a whole different level, which involves understanding how the immune cells could, and their product could play a role in terms of the ability of the brain to function as we age. And so, I mean, it opens up a whole new area of research, in my opinion. What do you think, actually, sort of having been here at the Nutrition Center for a few months now, how do you think nutrition could play a role here in terms of utilizing this information and coming up with ways that could benefit in terms of slowing down the loss of brain function with aging?
Right, and so that is a really important question, and makes me really happy that I am a part of your center, because I think nutrition will be pivotal in affecting the way now we can have healthy aging throughout our lifespan, especially with our nervous system, and now with this new sort of found immune system role. But I would defer to you, with your expertise in immunology and also nutrition, to ask, you know, can we incorporate discoveries like you and your collaborators have made on EGCG from green tea, the curcuminoids, other phytotherapeutics that can reduce inflammation. So what's elegant about this Science paper was that they actually did affect inflammation and showed improved cognition in mouse models of aging. And so what I think we have to ask now are, what sort of plant-based therapeutics would you suggest we should use in an aging mouse model to try to affect this system, which when it becomes inflamed, you don't learn as well?
Right, right. No, I think that's actually, I mean, we have a lot of information about what plant-based or other dietary components, the impact that it could have on cytokines that are inflammatory in nature.
Right.
We don't have much information specifically about how nutrition can impact the type 1 interferon cytokines. And I think that will be an area that will be quite interesting. There's some information not directly in the brain, but in other parts of the body. And I think what will be very interesting, and perhaps you and I can collaborate on, is to look and see whether in fact we could manipulate the whole type 1 interferon system in the brain to nutrition and then impact cognition.
I think that's going to be a really important experiment, Simin, and I think we should start it today. Where what I think we're going to find is that if it becomes dogma, which I believe it will, that aging brain and inflammation become sort of synonymous, then I believe that there are also studies that we are going to be involved in, that you well know, and some of the grant-related projects we've submitted, where we believe that it's sort of a mind-body issue of, does the brain affect the body aging? Does body aging affect the brain? And there's literature expanding now showing that in fact that does happen. So if we can reduce the inflammatory tone of the aged brain, and get the brain to be somewhat younger, as in this experiment, which they did with elegant parabolic experiments, where they put young blood into old animals and showed that they reduce inflammation and improve their learning. That fits with what I think we're seeing in the Alzheimer's world now, where there's quite a buzz about the potential for introducing young bone marrow-derived hematopoietic stem cells into elderly people who are at risk or actually experiencing cognitive decline, possibly associated with Alzheimer's. That young blood makes Alzheimer's better, young blood makes old brains seem younger. Wouldn't it be nice if we could use what you just suggested to reduce the tone of inflammation in the brain, and then consequentially get the body to be aging less?
No, I agree. I think those experiments will be fascinating to do. Thank you so much.